Now we're going to complete the DNA replication notes. So what is DNA? Well, DNA stands for deoxyribonucleic acid. So deoxyribonucleic acid is DNA, and that is stored genetic information. Store genetic information and instructions for making proteins. So DNA, deoxyribonucleic acid, store genetic information, instructions for making proteins. So before the cell divides, the DNA is copied in a process called DNA replication. DNA replication. DNA replication, copying of the DNA makes identical strands so that when they separate during anaphase and go to the opposite ends, we end up with two sister cells. They are identical. DNA replication makes identical copies of the DNA strands. During which part of interphase does this process occur? Okay, remember, interphase is not a part of mitosis, but it happens just before mitosis to allow the cell the opportunity to make more DNA and to make more organelles and to do all those things that it needs to grow and get ready to divide again. So during interphase, during what's called the S phase of interphase, is when DNA replication is going to happen. I think about S means synthesis, which means to make. So during the S phase, synthesis phase of interphase, then that is when the DNA will be copying the cell, will be making more DNA. So that is during the S phase of interphase. So DNA replication happens during the S phase of interphase. I'm going to go back and put that there too. DNA replication happens during the S phase of interphase. For the role of enzymes, remember the DNA is a double helix. It looks like kind of like a ladder. And going up the sides of each side of the ladder, we have sugar, phosphate, sugar, phosphate, sugar, phosphate up and down the sides of the ladder. And then the rungs, that's where your bases are. And those bases are held together by hydrogen bonds in the middle of that. So we have to have enzymes to come in and open it up so we can lay down complementary bases with those bases to make a copy of that DNA. So we have to have those enzymes come in and open up that DNA ladder, split it down the middle so that new bases can be attached to those bases down the middle and make a copy of the DNA. So the function of that helicase is to go in and open it up. It unzips the double helix. We refer to it as unzipping the double helix. And I'll see if I can find a picture of it in there too. So the function of helicase is to unzip the double helix. Real quick and see if I can find a picture of that. Okay. Shrink this a little bit where we can look at it better. Okay, so here's a picture, kind of like that DNA helicase working there. It's going to unzip the double helix. Now I see here, so double helix looks like a ladder, and right here, this is a section that has been opened up by DNA helicase, and then it's able to, let me just a little bigger so we can see. Don't know what's going on there, but okay. Anyway, it looks like a ladder here, a twisted ladder. And right here it's unzipped. It was the DNA helicase that came in here and unzipped the ladder, and now we see new bases being attached. So this will end up being a new copy of the DNA. It's laying down the complementary strands like was on this side. And then over here, there's new bases been added to this one. That's going to be a new strand of DNA. This was complementary like this one. So this strand will be one old and one new, and this strand will be one old and one new. And that'll be our copy DNA. So DNA helicase comes in and unzips it so those new bases can be laid down to make that new strand. DNA polymerase is actually going to put those bases into place, and it's going to proofread and make sure they're in there correctly. Occasionally errors do happen. Even though we have the polymerase proofreading, errors do happen, and sometimes that can cause problems. But the function of the DNA polymerase functions to attach the complementary bases DNA polymerase functions to attach the complementary bases and to proofread to make sure they're in the proper places. So DNA polymerase functions to attach the complementary bases and to proofread to make sure that they're inserted properly. So it's going to make sure your A's are matched to your T's, and your G's are matched to your C's, and your C's are matched to your G's, and your T's are matched to your A's. So this is the A to T, G to C part. We're adding those bases in there. Complementary bases being added to make, this will be a strand here, a new strand here, and then this will be another new strand right here. And they'll be identical. Okay, remember, polymerase puts in and proofreads, so the P's go together. That'll help you remember. Polymerase is going to put in and proofread. And the helicase unzips. Now, I think about maybe a suitcase with a zipper. You're unzipping. So helicase unzips the suitcase. Maybe that will help you remember that one. So replication in live cells. Prokaryotes have a single circular DNA molecule. Okay, so pro means no. So prokaryotes, those are bacteria cells. Remember, all prokaryotes that we know about are bacteria cells. So pro means no, and that's no nucleus and no membrane-bound organelles. There's no ERs and Golgis in prokaryotes. They're simple, that's why they can divide so quickly. So prokaryotes have a single circular DNA molecule in the, well, it's not in the nucleus because prokaryotes don't have a nucleus. So it is in the cytoplasm. It's just floating right out there in the cytoplasm. Sometimes it'll be in a little area that might be called a nucleoid region. But it's still just out in the cytoplasm. So prokaryote, the DNA is in the cytoplasm. Lots of times it's a ring that looks like this for bacteria. And it contains nearly all of the cell's genetic information. Replication in prokaryotes starts at a single point right here, and it's going to go that direction and in that direction until it meets, and then it's just going to come off as two. You'll have two rings then, two rings of the DNA that are identical to each other. Okay, not quite that simple in eukaryotes. Eukaryotes have much more complex cells, which means they're going to have to make lots more proteins and different kinds of proteins because they're more complex. So in eukaryotic cells, the DNA is found in U means true, U means do. They do have a nucleus. They do have a true nucleus. Okay. So in eukaryotes, the DNA is found in the nucleus. Patented chromosomes or chromatin. So DNA is on chromosomes, chromatin. They're often used interchangeably, DNA, chromosomes, chromatin. Replication may begin in a dozen or even hundreds of different places. So that helicase may come in and open that big, long strand of DNA. When it's stretched out, it's really long. And the helicase can go in and open it up in more than one place, sometimes even dozens of places, or maybe even hundreds of places. And it starts copying in both directions at the same time until they all run into each other, and then it comes off as two identical strands of DNA. Two identical double helixes. And there's a picture of kind of what it looks like. Helicase came in and opened up right here, and then starts copying in that direction and that direction at the same time. And also right here, it's copying in that direction, and here it's copying in that direction, until they all meet up, and you get two strands. And that blue right there, that was the original strand. This is the complementary strand that was laid down with that one. And then this was the original strand. That's a complementary strand. So now we have two identical strands of DNA. So telomeres. Telomeres are the ends of the DNA. And sometimes they're kind of complicated. They have a lot of repeats in there. So the telomeres are the ends of the DNA strands. Where there are lots of repeats. On a repeating basis, maybe. And this can make telomeres difficult to replicate. So the ends of the DNA molecules are difficult to replicate. Errors are more likely to happen. I guess because they're right there at the end, sometimes they come loose and tend to miss some of those base pairs. So the ends of the DNA in the molecule are difficult to replicate. So eukaryotic cells use a special enzyme called telomerase to help do that job. So telomerase is going to help. The function of the telomerase is to ensure that the telomeres are copied and are copied correctly. They try to ensure that it doesn't kind of fall off track there at the end and miss some of the bases. So it's to ensure the telomeres are copied and are copied correctly. So in rapidly dividing cells, the telomeres help to prevent genes from being damaged or lost during replication. The telomere is often switched off. Telomerase is often switched off during adult cells. In cancer cells, sometimes it becomes active again, and then that's part of what allows cancer cells to divide rapidly. Okay, so that's your notes. So that along with our PowerPoint that we went over. So take a look at those and study those for the test.